Hello, welcome to Biology, the scientific study of life. More about modern reproductive techniques. Amniosynthesis and corynecphilus sampling are invasive techniques in which amniotic fluid or fetal cells are obtained for genetic analysis. Non-invasive procedures usually are ultrasound imaging to detect fetal condition. A newer non-invasive technique relies on the fact that a few fetal blood cells leak across the placenta into the mother's bloodstream. A blood sample from the mother yields fetal cells that can be identified with specific antibodies which bind to proteins on the surface of fetal cells and then tested for genetic disorders. Diagnosing genetic diseases in fetuses poses ethical questions. To date, almost all detectable disorders remain untreated in the uterus, and many cannot be corrected even after birth. Parents may be faced with difficult decisions about whether to terminate a pregnancy or cope with a child who may have profound effects and a short life expectancy. These are complex issues that demand careful information or informed thought and competent counseling. Reproductive technology can help with a number of infertility problems. Hormone therapy will sometimes increase sperm or egg production, and surgery can correct disorders such as blockade of duct. Many infertile couples turn to fertilization procedures called assisted reproductive technology (ART). These procedures generally involved surgically removing eggs, secondary oocytes, from a woman's ovaries, following hormonal stimulation, fertilizing the eggs and returning them to the woman's body. Unused eggs, sperm, and embryos from such procedures can be frozen for later pregnancy attempts. With an vitro fertilization (IVF), the most common ART procedures, the oocytes are mixed with the sperm in culture dishes and incubated for several days to allow the fertilized eggs to start developing. When they have developed into embryos for at least 8 cells each, the embryos are carefully inserted into the woman's uterus and allowed to implant. In ZIFT zygote intrafallopian transfer eggs are also fertilized in vitro, but zygotes are transferred immediately to the woman's oviducts, fallopian tubes. In GIFT gamete intrafallopian transfer, the eggs are not fertilized in vitro. Instead, the eggs and sperm are placed in the women's oviducts in the hope that fertilization will occur there. Any of these procedures can use sperm or eggs from donors. These techniques are now performed in major medical centers throughout the world. Though they cost thousands of dollars per attempt, they have resulted in thousands of children. To date, evidence of abnormalities resulting from the procedures has been scanty. Animal development 47. A bodybuilding plan for animals. It is difficult to imagine that each of us begin life as a single cell about the size of the period and the end of this sentence. Notice that the size 1 milliliter shows a human embryo. About 6 to 8 weeks after conception, the brain is forming in the head, and the developing heart, the red spot in the center, this one, has already begun to pulsate. It takes a total of only about 9 months for a single-celled zygote or fertilized egg to be transfigured into a newborn human, built of trillions of differentiated cells organized into specialized tissues or organs. The question of how a zygote becomes an animal has been asked for centuries. As recently as the 18th century, the prevailing notion was formation, the idea that the egg of sperm contains an embryo, a performed miniature infant, or a homunculus that simply becomes larger during development. The competing explanation of embryonic development was epigenesis, originally proposed 2000 years early by Aristotle. According to epigenesis, the form of an animal emerges gradually from a relatively formless egg. As microscopy improved during the 19th century, biologists could see that embryos took shape in a series of progressive steps, and epigenesis displaced formation as the favored explanation among embryologists. The concept of performation may have some merit, however. Although an embryo's form emerges gradually as it develops, aspects of the developmental plan are already in place in the eggs of many species. An organism's development is determined by the genome of the zygote and also by differences that arise between early embryonic cells. These differences set the stage of the expression of different genes in different cells. In some species, early embryonic cells become different because the uneven distribution with the unfertilized egg of maternal substances affect development of the cells that inherit them during early mitotic divisions of the zygote. In other species, the initial differences between cells are due primarily to their location in embryonic regions with different characteristics. In most species, a combination of these two mechanisms establishes differences between early embryonic cells. As cell division continues and the embryo develops, mechanisms that selectively control gene expression lead to cell differentiation, the specialization of cells in their structure and function. The timely communication of instructions telling cells precisely what to do and when to do it occurs by cell signaling among different embryonic cells, along with cell division and differentiation. Development involves morphogenesis, the process by which an animal takes shape and the differentiated cells end up in the appropriate locations. By combining molecular genetics with the classical approaches to embryology, developmental biologists are now beginning to answer many questions about how a fertilized egg cell gives rise to a particular animal. After fertilization, embryonic development proceeds through cleavage, gastrulation, and organogenesis. Important events regulating development occur during fertilization, and each of three successive stages that begin to build the animal's body during the first stage, called cleavage cell division, creates a hollow ball of cells that, or the blastula from the zygote. The second stage, gastrulation, produces a three-layered embryo. The gastrula, the third stage, organogenesis, generate rudimentary organs from which adult structure grows. Fertilization, the sperm and egg gametes, which unite during fertilization and both highly specialized cell types, are produced by a complex series of developmental events in the testes and ovaries of the parents. The main function of fertilization is to combine haploid sets of chromosomes from two individuals into a single diploid cell. The zygote, another key function, is activation of the egg. Contact of the sperm with the egg surface initiates metabolic reactions with the egg that trigger the onset of embryonic development. Fertilization has been studied most extensively in sea urchins. Their gametes can simply be combined in seawater in the laboratory, and subsequent events are easily absorbed, although sea urchins, phylum Echinodermata, are not vertebrates or even chordates. They share with those two groups of characteristic of being deuterostomes. Despite differences in detailed fertilization and early development in sea urchins, provide good genome models for similar events in vertebrates. Acrosmal reaction. The eggs of sea urchins are fertilized externally after the animals release their gamets into the surrounding seawater. The jelly coat that surrounds the egg provides as a source of soluble molecules that attract the sperm, which swim toward the egg. When the head of a sea urchin sperm cell comes into contact with the jelly coat of a sea urchin egg, molecules present in the egg's coat trigger the acrosmal reaction, as shown in this figure. Notice that. This process begins when a specialized vesicle at the tip of a sperm called the acrosome discharges hydrolytic enzymes. These enzymes digest the jelly coat, enabling an elongating sperm structure called the acrosmal process to penetrate the jelly coat. Molecules of a protein in the tip of the acrosmal process then adhere to molecules of a specific receptor protein on the egg surface. These receptors extend from the plasma membrane of the egg cell through the vitelline layer, a meshwork of extracellular matrix molecules lying under the jelly coat in sea urchins and many other animals. This lock and key recognition of molecules ensures that egg will be fertilized only by sperm of the some species, such as specific, is especially important when fertilization occurs externally in water where gametes of other species are likely to be present. Contact of the tip of the acrosome with the egg membrane leads to the fusion of sperm and egg plasma membrane and the entry of the sperm nucleus into the cytoplasm. The egg either contact or fusion of the membrane causes ion channels to open the egg cell's plasma membrane, allowing sodium ions to flow into the egg cell and change the membrane potential. This change in membrane potential, called depolarization, is common among animal species, occurring within about 1 to 3 seconds after a sperm binds to an egg. Depolarization prevents additional sperm cells from fusing with the egg's plasma membrane. Without this fast block to polyspermy, multiple sperm could fertilize the egg, resulting in an apparent number of chromosomes in the zygote. Okay, thanks for listening.